Galaxies the whole 3 nights, in case you're wondering. Because someone asked. Sorry.
Good evening to everyone, and welcome to the first of the 3 Atoms to Galaxy nights brought to you by the Pint of Science Festival. I'm Claes. I'm a physics PhD student at the University of Edinburgh, and I study how DNA organizes within the cell nucleus and how this relates to the cell functioning. And I will be hosting tonight together with Jenny.
I'm also a PhD student at the University of Edinburgh, and like Claes, I study DNA, but we study DNA in very different ways. So I look at DNA if we take it out the cell, and I can fold it into different shapes. So this is a technique known as DNA origami, but Maria is going to explain a bit more on this tonight. So we have 2 amazing speakers tonight, Davide and Maria. So Davide is going to take us through DNA inside the cells. So you've probably heard a lot about DNA as a genetic storage information. However, you've probably not heard a lot about the tangled dynamics of DNA within the cell. So Davide is going to speak a bit about that. And then Maria is going to chat us through how the potential applications in the future for DNA to be used as a smart material. So I'll hand back to Claes to explain a bit more about how the evening's going to work.
So the talks will be 20 to 30 minutes long, and then there will be lots of time for questions. And then we'll have a short break of 20 minutes, during which you can grab another pint or join us for a short activity that we have organized. So without further ado, I guess our first speaker is Davide Micheletto. He's a researcher at the University of Edinburgh and our boss.
You gotta be nice.
And, so he studied physics in the University of Padova and then moved to... Yes.
Thank you. Hi, somebody ordered a margarita pizza. I was standing in front of him and that's why I couldn't see him.
I think it's fine.
First success of the night.
I think he's the 3rd speaker, right?
So as I was saying, Davide studied physics in Padova and then moved to Warwick to do his PhD in polymer physics, which basically means studying how spaghetti behave. A gem of his PhD was that he invented a novel type of pasta called annelloni, which is shaped as a ring-shaped spaghetti, which is possibly the most annoying type of pasta to eat. He now uses polymer physics to understand how DNA is stored in our bodies. Fun fact, when he was 16, at the peak of his football career, he played at a championship against Lionel Messi, and he remembers this very well because he was playing in defense and was supposed to mark Messi, and we remember very well because we've heard this story so many times. Let's give him a warm welcome.
Oh dear. So, is it okay if I speak loud? Yeah? Yeah? Want to go on the back? Fantastic. Let me put up the dog. Alright. Hi everyone. I'm very excited actually. Yes. Thanks a lot for coming. I was, at some point, when I saw, I could see the number of tickets being sold, and I was a bit worried at some point. But it's great that you're all here. So a quick poll actually, how many of you are kind of scientists or in academia or kind of studied science? So okay, the other way around, sorry. Who has not? Sorry. Who has not done that? That's fantastic. Beautiful, fantastic. Okay, so yeah, that's good. So this talk is going to be especially for you. So yeah, stop me if I say something that you don't understand, and I'll have a shot. I stole it a bit from Tyler. Okay, so my name is Davide Micheletto. I'm a reader at the University of Edinburgh. I'm not as serious of a boss. Anyway, I think, I don't know. And so my lab is called Topologically Active Polymers Lab, and it's actually funded by Europe. So thanks a lot, Boris. And so today I'm gonna try to tell you something about DNA that you didn't know, and hopefully when you go home, you know something more that you didn't know before, right? So first, before we get started, can I ask you all to stand up, please? Yeah, I know it's a lot to ask. If you reach up, okay, do you think you can reach 2 meters? Easy. Yeah. Right. So this is how much DNA you have in each of your cells. Can you believe that?
Yes.
Yes. Fantastic.
You should told me that before.
All right. So that was very easy to kind of convince you. I was hoping that someone would say no, and then is there someone who doesn't believe me? Because then, what? You shouldn't believe me just because I told you something. You should ask me, how do you know? And then I'll tell you the whole story. Anyway, so let's get started. So today I will talk you through basically the shortest segment of DNA that you have in your body, how it is packaged inside each of your cells, and then Maria here will tell you how you're using DNA to make this kind of. So this is a Mona Lisa, yes, made of DNA, and it's tiny, 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 very tiny. So Maria will tell you how we managed to do this. Right, so let's get started. What is DNA and where is DNA? So DNA is a sequence of letters that encodes the blueprint of life. So probably you heard this. So DNA, the structure of DNA is a double helix, which was discovered in 1953. And it is a series of letters. So it's A, T, C, and G. So they pair up in a very precise way, in the sense that A always pairs with T, and C always pairs with G. And because of this, basically each strand can copy the other strand. So when you've got the two strands here, and when they divide, one strand can be copied into another strand. And this is how basically life proliferates. The genome is the whole DNA that is inside your body. And my DNA, my genome is different from your genome, from your genome, from your genome, everyone's genome, because of a difference in the sequence of letters. And this is a very long piece of DNA. So in each of our cells, we have 6 billion letters. Okay, so that's a lot of that's a very sequence of letters, and this is the genome. Not all the DNA is equal, right? So there are some special bits of DNA that are called the genes, and the genes are the ones that encode actually for the proteins. How do they do that? So there are special machines, proteins, themselves proteins actually, that transcribe DNA into something called RNA. The RNA is then also translated by other proteins into a series of amino acids, and the amino acids fold in to form these proteins. And this is how basically cells produce insulin or muscles and so on. Right. So this is how it works, more or less, very quickly. This is the aim of this talk is not to tell you exactly how it works in terms of genetics, but the other thing that I want to say in this slide is where it is. So let's take a human body. Let's take us, right? Let's take your hand, have a look at your hand. Try to find the smallest feature that you can find on your hand. Smaller than that, there is a cell. You cannot see it because it's smaller than the smallest feature that you can see. And inside each of your, one cell is about 0.1, so a tenth of a millimeter. Inside you have a tenth of that is your nucleus, is a nucleus of a cell. And then inside the cell, there is about 10,000 times smaller, there is basically the DNA. The DNA, which is 2 meters, and it's all packed in there. But the thickness of this object is 10,000 times smaller than the nucleus. Right, so you might think that DNA is a thing for biologists, right? Okay, but Max Delbruck studied astrophysics. He won a Nobel Prize in medicine for his studies on DNA. He started the whole field of molecular biology in 1920s, 30s. Erwin Schrödinger, very famous quantum physicist. Probably if you watched The Big Bang Theory, you know him because of the Schrödinger's cat. Do you guys know Schrödinger's cat? I hope you do. And he actually had a series of lectures on what is life before actually DNA was discovered. And Francis Crick, one of the two guys who discovered the structure of DNA. And he was a physicist, and he won a Nobel Prize in medicine. And so physicists were there since the beginning, right? And we're still are. Anyway, let's get a little bit more precise. So DNA inside us, let's go get some numbers. So 1.75 meters is about the average height of a person, let's say. you got, as I told you before, you have a fraction of a millimeter, so a tenth of a millimeter and a tenth of that, so a hundredth of a millimeter, you have of a nucleus, and inside you have 2 meters of DNA. So I'm going to tell you this number so many times that when you go home, you're gonna dream about it. So 2 meters. And now you're gonna help me with some math. So in each body there is, look at that animation. That is good. 30 trillion cells. Okay, 30 trillion cells times 2 meters is 60 billion kilometers. Can you wonder a little bit what is the size of 60 billion kilometers? The distance between Sun and Neptune is about 4.5 billion kilometers. So what this means is that, and this is not in scale, so the distance between Sun and the end of the solar system, you can span that with the DNA inside each of our body, not the whole humanity or each of our bodies, many times. Okay? So this is pretty cool, isn't it? Yeah. Anyway, okay, fantastic. So let's then you should ask me, wow, that's fantastic, right? But then how is it packaged inside our cells? Okay, I'm going to tell you that. So, or also as I call it, how IKEA got arrived second. So this is why. So this is 2 meters in 0.02 millimeters. Okay, this is basically a packing fraction or a packing efficiency, if you allow me this a little bit technical terms, of 100,000 times. So if I compare it to an IKEA kitchen, basically it's like IKEA sends you the kitchen in like a spot that is this size. And I had to do the fireworks because it's so small that otherwise you will not be able to see it. And so imagine that. I mean, that would be pretty cool. But the problem is, how do you figure out how to build a kitchen? You need pretty good instructions, which are much better than the ones that IKEA sends. Right? So, at least this is. Anyway. So, right. So the way it is packaged is in a hierarchical way. Okay. So you have your DNA. So this is the double helix in a very artistic way. So right. And then the double strand of the DNA is wrapped around objects that are called histones. And so this is I want to show you a real image, right? Because this is me sketching it, right? But then you shouldn't believe me just because I tell you. So, and here I'm showing you a real picture from someone who did the experiment. And so this is the gray stuff here is the DNA double helix. I hope it's a bit small, but yeah. Anyway, I hope you back there you see it. And then the colored stuff is the protein. It's a protein called the histone octamer, where the DNA is wrapped around. And then the histones, so this object here is called the nucleosome, and the nucleosomes come together in a fiber. Okay, these are beautiful structures of chromatin of arrays of nucleosomes that come together in something that is called a chromatin fiber. Okay, and then chromatin is folded. So this green string is basically representing the fiber of chromatin that is all folded up in a way that I'm going to tell you in just one slide. I know that you're all kind of wait to hear it. And then it's all packaged inside this little nucleus that I told you before. So how are the chromosomes packaged? Well, so at some point scientists had this very beautiful theory. So there are two ways, let's say, that chromosomes are packaged inside the nucleus. They're either neatly folded, like my drawers, obviously, or they are kind of a messy and kind of packaged randomly as my wife's drawers. Do you believe that? No, not personally. Right. So then basically there was a pair of very smart scientists that said, okay, so let's imagine that we can color the chromosomes inside our cell, and then we can see whether the colors are all scrambled up, they're all mixed up, or they occupy different positions in the nucleus. And so this is the picture that they produced in 2001. So this is not your eyes that are getting, because of too much beer, but it's actually, the picture is blurry because it's 2001, and it's 20 years ago. So this is a good image for a nucleus that is 100 times smaller than a millimeter. But what you should notice is that the colors do not mix. So each chromosome, and we have 46 chromosomes inside these cells, and the colors don't mix. So you see that you got red there, purple there, green there, yellow there, but you don't see gray everywhere. You kind of see different regions, they have different colors. And so how do you explain this? So basically what they came up with a conclusion, so the experiment, they had an hypothesis, and then they did the experiment, and then what they figured out is that actually it is folded very neatly and also by color. It's it's very smart the way chromosomes are folded. But then you should ask yourself, how the hell is possible, right? Because there isn't like a little human or a little thinking person that is there folding the clothes like I do at home, right? So there comes another physicist. In 1993, a very smart guy, let's say that, named Shura Glossberg, so proposed this concept. So let's say that you have a very long string that is from blue, is colored from blue to red, okay? Now, how do you package this very long string inside a sphere? So you go several ways you can do that. One way is you just cram it, you just put it in as strong as you can, as quick as you can, just random, and you get something like this. So colors don't match, they're all mixed up. Here you go red, here you go blue, then you go blue again, you go red again, and yellow again. So it's all mixed up, right? So this is more or less like the randomly packaged drawers that I was showing there. This is not good. This is not a good model. What is another model to package my string inside the spherical space is by using a fractal curve, a fractal space filling curve, almost like your snowflakes or Romanesco broccoli. So if you ever, apparently some people here actually in my group have never had Romanesco broccoli. That is appalling. Who was that? I can't remember. But anyway, but what you should notice is that by folding it in a fractal structure, so a fractal means that it's self-similar, no matter where you look, how closely you look, they always look the same. Then basically what happens is that the colors are not mixing. So you've got red there, yellow there, blue there, and green there, right? So they're not mixing, and this picture very closely resembles what the nucleus actually looks like in the experiment. And so one of the findings was of this story is that chromosomes inside our body are fractal. Would you have guessed that? The chromosomes in our body, in our cells, look like Romanesco broccoli. Did you ever guess that? I did. So in spite of this, so this is true for healthy cells. Diseased cells have a different organization, so it's not as neatly organized. They're not as neatly organized, and this is why it's important, basically, what we do in terms of what we study. So Claes was saying that she studies how the DNA is organized, and this is important because the better we understand how the DNA is organized inside the cells, the better we understand also the cell health and the diseases associated with the misorganization of the DNA. Now, again, you got a question?
Yeah.
Is it hard?
Yes, it is.
Oh dear.
I'm a chemist by training, so I have
Oh
no
dear,
idea.
the worst kind.
Just because you're a physicist.
Sorry.
Right, if you go to the slide before, you say about fractals, and
Yeah.
when you look at the fractal at any distance, it has the same shape.
Yeah.
When I look at the red spot, when I zoom to the red spot, I will see more of the red spot. Why? Why then is a fractal?
So the way it is folded, so if you look at the way it is folded, the piano curve, it's a self-similar object in the sense that it is folded, a big block is folded like a small block. At some point, you get so small that you cannot do that anymore because a fractal is a mathematical object that is kind of abstract. Then when you put it in a physical terms, you have a finite length at some point. You have one nucleosome, one base pair. You cannot go smaller than that. But if you zoom out enough, you see that there is a repeated hierarchical way in which it is folded.
But we've got to ignore the color.
That's my question.
Oh, I see what you mean. So with this, what I mean is that the red stuff will stay close to the red stuff, and the yellow stuff will stay close to the red stuff if you fold it like this, in a self-similar way.
But it's the shape that's fractal, not the colors.
Yeah, that's true. Okay, I missed the question. That was a hard question. But glad that we sorted it out. Okay, fantastic. Now, so you should ask me why. How do you know, right? How do you know? Is that because we can see it nowadays? Actually, so I showed you a 2001 picture. How am I doing with time, by the way? Because I think I'm running. Anyway, so one of the things that we can now do since 2012 is that we can really see chromosomes. So before I was showing you like this blurry picture, but now we can see them much better. So in 2012, there was this lady here, Zhuang, from the States, and she invented this beautiful microscopy technique that is quite a mouthful. It's stochastic optical reconstruction microscopy. It's quite, it's a lot of jargon, but the acronym is one of the best you'll ever see. It's called STORM. This microscopy is called STORM. Beautiful. Anyway, so so this was the best picture we could get of a brain cell of a neuron in 2012. 2013, after using STORM, this was the picture. So this is a major breakthrough in how we do biophysics, biology. So we can really see structures now at a super resolved structure, super resolution. And just give me 2 minutes to tell you how actually this technique works, because it's quite beautiful. So let's say that you see this kind of movie. You see a bunch of flashes. You don't know where they come from. Right? If you look at each frame of this movie, you don't know what's the structure, what's underneath that structure that emits these flashes. But if you put a spot on all the flashes that you see, and you kind of cumulatively take into account all the flashes, at the end, you figure out that you're looking at the Eiffel Tower. Right? So this is how basically this technique works. And I was kind of lucky enough that I could actually put my hands on one of these microscopes, STORM microscopes. They are 1 million pounds. So yeah, you have to treat it gently. And I was at the Western General Hospital, just across town, at the Institute of Genetics and Cancer. And so this is actually an experiment with human cells that I was doing. And so this is my computer where things are blinking, like in the image over here. And this was my microscope, which I was not allowed really to look inside here, because here basically there are very strong lasers that are hitting the cells to make the cells blink, basically. And yeah, I wasn't, so do not do that, but anyway, I did it for you guys. And so since then we could see amazing structures. So this is an image of chromatin in 2016. So you remember this array of nucleosomes that I was telling you about before? This is the same image. This is the image before STORM, and this is the image after STORM. And then we can even see RNA. This is a picture that I took, actually. I'm quite proud of this. Of RNA. So you remember the blurry picture that we saw before? This is how we see things now. So this was a major breakthrough that is pushing the field to the next level. Now, give me another couple of minutes, or maybe 5 minutes. And I hope you're still with me. So I want to tell you about this protein because this is my favorite protein. It's a protein that I love. Like if I had like a little stuffed toy, that would be. So topoisomerase is the smartest protein that there is because of the following reason. Even if the chromosomes are arranged in a fractal structure, I know that you still don't believe me, but it's good. There's still 2 meters of DNA in each nucleus, so it's going to be very, very entangled anyway. So imagine that you have a cell here. In purple is DNA, and the blue stuff is microtubules that are helping the nucleus to divide into the 2 daughter cells. Now, what you can imagine is that at this moment here, when the DNA compacts and then is split into the 2 daughter cells, at that point you have so much DNA there that there is going to be, forget about the headphones in your pocket. So this is far more complicated in terms of topology and messiness that you're going to get, right? So how do you solve this? Obviously, you cannot go there and undo. What do you do when you get a knot in your headphones? You kind of undo very patiently, but you cannot do that, right? I mean, this is what I do, and I get very frustrated and throw away the headphones. But nowadays, we don't have this problem anymore. So, yeah. Next year, I will not be able to do this joke anymore. Anyway, so nature has evolved basically a protein that solves this problem in a very elegant way. So there you go. This is my favorite protein, topoisomerase. It acts as a little scissor and a glue at the same time. It's a little scissor and it's a glue for the DNA. So what it does is every time it finds an entanglement, so something like this between 2 DNA strands, if you pull on these 2 things, it will break. You don't want DNA to break. That would be very bad for your cell. The cell would die, most likely. So topoisomerase finds it, cuts it, passes the one strand through the other, and then seals it back. This is almost like magic, although it's not magic, it's science, but to me it's almost magic. And this is how you get things solved and how you get a division. If you knock out, knockout is a very super jargon word. If you remove topoisomerase from your cells die. DNA gets broken in thousands of pieces, and you cannot do anything with it. And why is it important that we study it? Because there are some, in cancer cells, topoisomerase is very highly present. So there is a lot more topoisomerase in cancer cells. And this is because cancer cells divide very quickly. So there has to be much more disentanglement going on. So there must be much more of this protein resolving these knots. And this drug here, called the etoposide, actually prevents topoisomerase to act. And so by understanding how topoisomerase acts, we developed a drug, not me, but developed a drug to stop topoisomerase from acting and therefore slowing down cancers. So this is why it's also important that we study these things. Right, very final thing. Well, how am I doing with time? Sorry. Fine? Okay, you go.
Just a quick question. I was just wondering, the blue guy, presumably, does he join back immediately after he passes the red guy?
Yeah.
And how fast? That's a chemical question.
Binding affinity and everything. How fast?
Well, you can start from telling like this email, that's video that you
I'll have to think about it. So a cell division occurs in the range of 1 minute.
So the video that I'm looking at right now, how much was it speeded up? 10 minutes.
10 minutes is a cell division, more or less.
Okay.
But this has to act much quicker. So the way, I'll tell you later. This is never going to end. But yeah, happy that there actually have so many questions. So the genome. So let me tell you this final gem of the genome, because this is really beautiful. So what you're looking at. So what I'm going to try to argue here, I'm going to try to convince you. You're going to have to believe me. That the genome is a smart Swiss knife, so it can do many things at the same and in different ways, as follows. Okay. So what you're watching is an embryonic stem cell. So it's the first cell when sperm and egg meet to generate a new organism. This is actually a salamander, and this is a movie actually that I recommend you to go on YouTube and watch because it's beautiful. So the one cell become 2, become 4. Go to 8. I'm gonna let you watch. It's not too long. It's like 10 seconds. So. And then at the end, when it has split so many times, I make a jump in the movie, because otherwise it's 3 days long, and obviously you don't want to stay here. At the end, you get a full salamander, right? So now you should stop. Did you stop? Thank you. So what you should have appreciated is that everything started from a single cell. So the DNA that is inside the salamander's eye and the DNA that is inside the salamander tail have to be the same, because they were all copied from one cell, right? Are you convinced on this? So how the hell is possible that one cell becomes a part of the eye and one cell is becoming part of the tail or part of the heart or part of whatever else, right? So we're gonna try to understand this, how this happens. Another very smart guy, Conrad Waddington, here in Edinburgh, 1945, so before DNA was discovered, he proposed the following model. Here I have my stem cell, right? It's up on the valley here, and then this basically is rolling down. This is my embryonic stem cell, and it's basically rolling down. And depending on the choices that he makes, it can either become an eye or become a tail. How? Okay, so this is the general concept. I haven't told you exactly how to do that. Doesn't make the choice. But basically, what happens is that, do you remember the little spheres that I told you before about the nucleosomes? So the nucleosomes is where the histones is where the DNA is wrapped around. on top of the histones, there are little tags that are called, basically they are biochemical marks that identify one nucleosome from another, okay? And so basically what I'm trying to convince you is that the eye cell, if you stretch the genome and you look at the different colors or biochemical marks that you have along the genome, along the series of nucleosomes, this is going to have one pattern. By the way, this is also known as epigenetics. So this is let's say this is the patterns of colors on the nucleosomes in the eye, let's say, just for example. And this is the pattern of colors that you have in the tail. And then the other thing that you have to to join the dots is that a red color somewhere means that this piece of DNA that is wrapped nearby that is being transcribed. So it's a gene that is on, that actually can make proteins. And this is instead blue if it is a gene that is off. So basically this is a sort of a crossing light system. So in the sense that when it's red, it's the other way around. It's the other way around to the normal one. But anyway, so when it's red, you can actually transcribe. So the proteins are actually transcribe the gene. The gene is on, so it's firing. It's making proteins. But when it's off, when it's blue, it's off. Okay. And depending on the pattern, you can turn on and off different genes. So let's say that you have 20,000 genes in your genome. Only a fraction of those will be on, and others will be off, depending on these choices that you make, that the cell is making, going down this valley. Now, this is my final slide, I swear. How do we model this as a physicist? How does this link back to physics? So what I've done actually in 5 years ago was to try to model this decision making with a simple model, as physicists do. So I was starting from you saw this random string. I'll go back just a second. So this random string in space, which was completely randomly colored. And I simply assign some rules so that red colors like to stay with red colors and blue colors like to stay with red colors. And I could simulate basically a sort of a self-organized choice that the polymer, the string, was becoming compacted and all colored the same way. It was all becoming red. Okay? And it turns out, so this is in some sense how we model choices of how the cells are making choices. And it turns out, this is a big jump, so I don't expect you to kind of say, oh yes, I can see that. But basically, why there is a water kind of pot there, boiling pot, is basically because the transition that we see from the disordered state where the cell didn't know what to do to the state where the cell wanted to become an eye or a tail or whatever, we are modeling as a so-called first order phase transition. That is the same way that water boiling is modeled as. Okay, so basically what we are in some sense saying as a physicist, we are modeling the cell fate decision of how cells decide their fate by making a model that is similar to that of boiling water. this is the essence of it. Anyway, okay, so I'll stop here, I swear. Take home messages. How long is the DNA in each of your cells? 2 years. Thank you. Then the other keyword that you should remember is that it's hierarchical. IKEA, better than IKEA. It's a fractal, although she doesn't believe me, but then I'm going to sort it out. Even if it is a fractal, it has knots, right? And the knots are resolved by my favorite protein, which is called... Fantastic! You were listening. That advances in microscopy really make the difference. Really make a difference. Making... Hold on. And then... And the cell fate can be modeled as a boiling pot of water. Okay. What is next? There are a lot of things that we don't know exactly how topoisomerase works, actually. We don't know exactly how it works. So there are a lot of people still studying exactly how he finds the entanglement and he can do the stuff that he does. And also about the cell fate, the model clearly that I showed you is very crude, right? It was very simplified, but that was a first step towards a full understanding of that. Okay? And then what's next, what's really exciting for me, is taking all these, which is arguably very smart, the way the genome is folded, to outside the cell. How we can make use of all this stuff to make actually materials that could be useful for us. Right? And that's me. you. I think I went over time a lot. What was it?
So we have around 10 minutes for questions.
I remember the rule that told you. No, I have a question. So, yeah, yes.
I have a question about the storm technique.
Yeah.
It sounds a bit like it's long exposure photography, but for like small things.
Yeah.
What exactly is it taking a picture of, and how do you control for noise?
Very good. So do you remember that I told you that it's a stochastical reconstruction? So what happens is that there are some special fluorophores that blink. So here I wasn't telling you the whole truth because I had to cheat a little bit. But basically the fact that it's stochastic means that imagine that in the Eiffel Tower all of the flashes are on. You cannot really tell one flash from another. But if you stochastically turn them on, you can see a subset of those. Now, light, you cannot see light. So a spot smaller than 200 nanometers will make basically a blob of light that is at least 200 nanometers. And this is because it's the resolution of light. This is Abbe limit. So in order to find, so if you have 200 nanometer blobs, let's say that you have 10 nanometer objects that are emitting light that are closer than 200 nanometers, then you cannot separate this blob from this blob because the blob is bigger than the distance. But in the moment that you turn one on and the other off, and then you turn the other on and the other off, right? Then you can say, oh, this is one spot and this is the other spot. And then you localize them. You say, oh, this is where this spot is, this is where this spot is, and you reconstruct it like that. Does it answer the question?
Yeah.
It's good. Fantastic. Other questions? Mm-hmm.
You have two very interesting but different videos of when the cells are splitting.
Mm-hmm.
The case in the egg and then the other case with something else.
Yeah.